Hello everyone, welcome to week number 3 in Biology 120. This week is all about cells, including bacterial cells versus our cells. And we're going to get into that in just a second. So last week was all about the chemistry of life. You learned about the macromolecules, the importance of carbohydrates, how they carry lots of energy. Some of them are used structurally. Lipids, fats, oils, waxes, used also to store energy, also used as hormones, which are chemical messengers. There are other functions. They serve as part of the plasma membrane for cells, the phospholipids, which create a barrier between the inside of the cell and the outside. Proteins, most proteins are enzymes. Enzymes run all the biochemistry in cells. So they're terribly, terribly important. And very specific ones have very specific jobs. So that's the importance of proteins. And then we had nucleic acids. Many of those are important in heredity and passing along genetic information into the future generations, but also there's some of the energy molecules like ATP that are nucleic acids. So you learned about that. You learned about the importance of water, and water, as far as life goes, water is in every single cell that there is, so it has to be extremely important. One major function of water is that it absorbs heat from all the chemistry that's going on in cells. Also, you learn the importance of the element carbon, and carbon is so important in the backbone of all organisms, molecules, because it can form 4 bonds with other atoms or molecules, and therefore you can make highly complex, huge macromolecules based on the element carbon. So that's why that is important. So that gives us a little chemistry background to help us work our way into biology. And so this week is all about cells and types of organisms. The first part of the work this week is about classification. Classification is just putting things into categories. And then when you say the category, then you understand characteristics about that one particular organism. And that's important so that we don't have to relearn everything over and over and over again. We naturally classify, and so do many other animals, right? Have you ever seen those funny YouTube videos with cats and cucumbers? They are hilarious, right? You got a cat and you throw down a cucumber. What do they think it is? They think it's a snake, and they jump up and run away. Yeah. That's classification. So that cat thinks that anything that's long and skinny like that is possibly a snake and possibly danger. So classification is just a natural thing that we do. And so we do this with organisms to help us understand them without having to relearn what everything is every time we come across something new. You've got some chapters to read. I suggest skim them. Read chapters 20, the first section. The first section in chapter 20 is all about classification so that you understand classification a bit better. There are three domains in life. Archaea are the archaebacteria, bacteria. Eubacteria are the true bacteria. EU means true bacteria. So the 2 domains out of the 3 in life are bacteria. And the last one is eukarya, and that includes the eukaryotes, which we are part of. Okay, that's important in just a second. So all of life is broken down into 3 domains, and 2 of those are bacteria, and the third one includes us. All right, then chapter 4. Skim chapter 4. Chapter 4 is about cells and cell structures. Cell structures are going to be important for part of your work this week. Read the Module 3 overview and watch the videos, and then I have a couple of extra ones, which I will tell you about right here. One of them is how antibiotics work. I will put this in the announcement so that you can just click on it, because that will help you understand part of the work that you have to do this week. So, say you've got a bacterial infection. Antibiotics only work against bacteria. They do not work against viruses or fungi or anything else, just bacteria. Okay, so you got a bacterial infection and your doctor prescribes an antibiotic. An antibiotic is a chemical that's going to kill the bacteria. Well, we got a little problem there because bacteria are made up of cells and our bodies are made up of cells. So why don't our cells die too? Okay, that's a really important concept. There's got to be some differences between bacterial cells and our cells that antibiotics can target, and that's exactly how they work. So watch that video to help you with some of the answers coming up. I also want you to watch this additional video. This is just the most amazing experiment that I've seen in decades. It is a Harvard study about antibiotic resistance and how fast it can work. And what they did was they set up a gigantic agar plate. Basically, it was a rectangle full of agar, and then they inoculated the edges of the agar, and they had different concentrations of antibiotic throughout the agar. Watch what happens. Watch how fast E. coli becomes resistant to 1,000 times the normal dose of an antibiotic that it is normally sensitive to. This is an amazing video. And this can happen in your body when you're taking antibiotics. So this is a really important experiment. I love this one. So please do watch that. It will give you some insight into how antibiotic resistance works. All right, here's one of my new pictures from Africa. Sorry, here I am going back and forth. I can't even keep it on a slide. Anyway, here's one of my new photographs from Africa. This is a white-fronted bee-eater. Is that not a beautiful, beautiful bird? Yes, it is a beautiful bird. Anyway, let me get back to what we're doing. This week, what do you have going on? Discussion board postings. Oh, look. Here's some stuff that you have to post about antibiotics that I was just telling you about. So you're going to answer the question, why do antibiotics target bacterial cells and not our cells? I just explained there's got to be differences in our cell structures that antibiotics target. If we have exactly the same cell structures, then the antibiotics are going to kill our cells too, and that is not good. We don't want that. So cell structures are important. Look at cell walls. We don't even have cell walls. We've got animal cells. We don't have cell walls. We have cell membranes. Cell walls. Ribosomes, which are where proteins are made. Take a look at that. There are also other differences in pathways that bacteria have that we don't. So, for example, bacteria can make folic acid, which you guys have heard about because we have to get that in our diets. Well, they make their own and we don't, so we can target that pathway. So there are some differences between our cells. Take a look at that. And then also you're going to answer the question, how our use of antibiotics increases antibiotic resistance. I know of someone right now, an elderly gentleman who had a knee replacement, and he was diagnosed with a MRSA infection at the site where they put in the knee replacement. He has had horrific trouble. He's been on IV antibiotics for many weeks now. They're likely going to have to go in and completely change out the knee replacement. This is not something good. I was talking with my sister about this, and I said, if it were me, I would take this very seriously and do something about it, because this is extremely dangerous to have MRSA, methicillin-resistant Staphylococcus aureus, in your body. And it is throughout his body now. Yeah, so it's very dangerous. So you're going to talk about that. And in your response post to your fellow students, you are going to discuss antibiotic use in livestock and should this be used. Let me preface this by what has been happening throughout history. So back in around the 1950s, 60s, farmers discovered, especially poultry farmers, discovered that when they fed their chickens and their turkeys, etc., feed that had antibiotics in them, that the chickens laid more eggs, that they produced more meat. So the farmers had a greater profit margin, right? These were not sick chickens, turkeys. No, they were not sick. So what the heck is going on? Why wouldn't you give them antibiotics and they're not sick? Are they producing better? Because of what's called silent infections. We are all fighting off silent infections. And those chickens and turkeys, etc., had silent infections. And so if you take away that stress on that organism's body, then they can do more production, like produce more eggs and make more meat for sale. So should we be using antibiotics in livestock for a reason like this? And the answer, of course, is no, because the more you expose livestock to the antibiotics, the more resistant all the bacteria out there in the world that are exposed to them are becoming. And some of them cross over into human beings. And so this is happening faster and faster and faster in the world, this antibiotic resistance, and it is one of the top 10 public health issues in the world today, antibiotic resistance in bacteria. Because we're getting to the point with some species of bacteria that we aren't going to have anything in our arsenal to treat them with. Getting to that point. So take a look at that. So that's your discussion board postings this week. Remember, initial post is due by Thursday. The 2 response posts are due by Sunday. Then you're going to begin your final project. You got to choose your topic. If you're not choosing one out of the list, you have to email me to get approval. This is a requirement because I've seen some wacky topics that people choose that don't have anything to do with biology, and it's got to have something to do with biology. All right, then you're going to follow the Milestone 1 guidelines and rubric. In the very beginning of this course, I told you that guidelines are the instructions, right? They're just the directions, the instructions to tell you what to do. The rubric is how this is going to be graded. That's all those mean, okay? Use the correct format. Outline with details. This is not an essay. Outline with details, and I'm going to cover that again in a second. Label your sections. This comes in outline form, and this is going to be important because you're going to add slides, and you're going to make a slide presentation out of this at the end. And so you need to know what to include on the different slides, and that's where the outline is going to come in handy. All right, let's see if I can go to the next slide. Hey, I did it for right now. So I just screenshot Milestone 1 guidelines and rubric. There's the list of topics. I went over this last week, Population ecology, air pollution, water pollution, global warming. Here are your topics. I gave you a little bit of a rundown last week in my video, so go back to that if you want to hear more about those, or ask me. Goodness, all you have to do is email me, and I will be glad to talk to you about any of these because I love them all. I gave you some other choices like pollution, plastic pollution, and then there's plenty more. So if you don't find something on the list, I can help you with it. I'll guarantee you. I got all kinds of other ones. All right. Now, specifically, what I want you to look at is the bottom part of this. Here are the instructions. So you see the outline format. Roman numeral 1, introduction. This should say this on your paper. Roman numeral 1, introduction. Now, what does it say to do? Describe the topic you selected. There's a very important word there. It says describe. So if you're going to describe genetic modification, you have to tell the reader what that is. You don't just say genetic modification is modifying genes. No, that's not helpful. Describe. Describe. Very important. Why is it significant or interesting to you? Why did you pick it? Why are you interested in this? Why is it significant or interesting to the general field of biology and the world itself, right? Support your response with real life examples. And that's pretty easy to do with any of these topics. All right, then the second section, Roman numeral 2, topic. The subcategories under this. A, where. Include these headings. I cannot express that enough. This is not an essay. This is an outline where you are describing stuff and you are answering questions. Describe the primary locational sources, including ecosystems and biomes, where the topic happens in the environment, right? So if we're talking about global warming, that's the biome. That's everywhere in the planet where you will find anything living, right? Which is totally different from population ecology. And say you're interested in the population of deer in your area, right? So population is members of the same species that live together at a particular time. So this is a very limited definition of species. They live together in a particular environment at a particular time, and they live together. That's a population. So that's totally different from a biome, right? How? How does this happen? Analyze the processes. How this happens. Who? Which species are involved, and which ones are affected by this, and who benefits from this? Are there individuals that benefit from this, including people in the human population, including businesses and industries and governmental entities and all that sort of thing? Who benefits from this? So that's what you got to include in this first week's milestone 1. This is the first part of your final project. If you have any questions, please, all you have to do is email me. I have no idea why that was blank. I have no idea what it's doing right now. Oh my word. Let's see. Well, got no idea what's going on. All right, my tip of the week. No idea. Okay, tip of the week. Put your assignments in the correct format. I was just talking about that. Create an outline, section 1, section 2, the subheadings. There are other specifics about this. This is a 1 or 2 page document in Word. I don't care about the font. Don't make it little. Don't make it big. Don't make it crazy. Just make it normal and normal margins. And don't make everything center justified. Make it left justified so it's easy to read. You know how to do it. And use in-text citations. Now, I got no idea why I'm on the screen. So anyway, I will just close right here. With that being said, I hope that you have a wonderful week number 3. And if you have any questions, because you may have them this week. Look at this. Even my notifications are on there. Oh well. If you have any questions whatsoever, please email me m.sigmon@snhu.edu. I'll be glad to answer